So we've all spoken here now about the watershed year that was 2017 with our 2 cell-based therapy approvals, Chimera for ALL and Yescarta for DLBCL. There are various reports that came out of ASH this year that were very exciting, including very encouraging data from Celgene and Bluebird related to their anti-BCMA CAR to treat multiple myeloma. And there's also encouraging progress from Juno and Adaptimmune and many other clinical stage companies working with TCRs, NK cells, gamma delta cells, multiple adoptive cell therapy platforms, and vaccines targeting neoantigen delivery. Our theme this morning will focus on the clinical outlook, regulatory and manufacturing supply chain partnerships, and ultimately market access. Each panelist will introduce himself, describe the company's core clinical programs, and provide a perspective on clinical progress anticipated in the next 1 to 3 years with the therapeutic platforms they are developing. They will then mention key issue or challenge they expect to address in moving towards commercialization, and finally, how they may expect to approach partnerships. They've got 3 to 4 minutes each to do that, and then we'll have a discussion. Okay. Thank you.
Hi, I'm Manuel, CEO of Mustang Bio, and we have 6 programs that we've licensed in from City of Hope, a comprehensive cancer center, as well as Fred Hutch Cancer Research Center. There are 3 that are in the clinic today. 2 have shown activity that has been publicly disclosed. One is the IL-13 receptor alpha-2 directed CAR-T for glioblastoma, which was the subject of a case report in the New England Journal in December of 2016. The second is CD123 for AML, as well as for the BDCN, which was the subject of an ASH presentation just a few weeks ago. And then CD20 at the Fred Hutch for B-cell malignancies, which is now open to accrual. The 3 preclinical programs should enter the clinic within the next 18 months at City of Hope. They'll file their INDs. These are all so far under City of Hope or Fred Hutch INDs. CS1 for multiple myeloma, HER2 for glioblastoma and metastatic breast cancer to brain, and PSCA prostate stem cell antigen for prostate and pancreatic cancers. I think I've already alluded to the progress we expect to make in the next year with respect to the 3 preclinical programs. With respect to clinical, I think it's important to note that we have leased our own manufacturing facility in Worcester, Massachusetts, and so that will give us the capabilities for cell processing, and we hope to file our first IND for CD123 in December of this year. In terms of challenges, I think that manufacturing is always a challenge, so we're going to speak about that, I guess, a little bit more later. I think the second challenge is solid tumors, and we were solidly invested in that space, and you've seen the deal we've done for CRISPR, and we hope to leverage additional outside technologies to strengthen the program and hopefully leapfrog us with respect to our ability to impact on the tumor microenvironment, on trafficking of T cells into tumors, and all of the other challenges that have been problematic in terms of the solid tumor space. And then finally, I guess the third challenge would be kind of a little bit the embarrassment of riches for a relatively small company, and that kind of leads us to partnering. We're right now not seeking partnerships. However, it's obvious that with the financing we have and the financing we expect to have in the future as these programs expand out, as we enter into combination therapies, phase 1B therapies with solid tumors, with additional combinatorial strategies, we will most likely need to seek partnerships, and we'll do that at the appropriate time.
Thank you, Manny.
Is that 3 minutes?
I don't know. I don't have my watch. I trust you.
Good morning, everybody. My name is James Noble. I'm the CEO of Adaptimmune. We do one thing. We make SPEAR T cells, which are autologous TCR high-affinity T cell receptor T cells for treatment of solid tumors. We have 4 INDs open, one of which was recently optioned by GSK. They took up their option late last year. We had great data in a solid tumor, which we presented at ASCO last year in synovial sarcoma. Effectively, the program was pivotal ready, and GSK took over the option. They're continuing some of the trials, obviously, and we are transitioning that program. But today actually marks a watershed for us because we announced the first data from our wholly owned programs. We have 3 other INDs, all wholly owned, targeting 3 different antigens called MAGE-A10, MAGE-A4, and AFP. And we put out the first safety data today showing that the MAGE-A10 construct after 8 patients is not cross-reactive at all. So we are now able to go to therapeutic doses. So it's a bit of a watershed day for us, announcing our first ever data on our first wholly owned program. As I say, we are actually in around 20 cancer centers in the United States. We are in 8 different solid tumors with 3 different programs, and they will all report out over the next 1 to 3 years, the time frame that Bob set us. It is also, mentioning manufacturing, we put out 2 announcements this morning. If you want to get manufacturing right in autologous cell therapy, you have to get your vector supply right. There's been a lot of discussion about vector and the inability of people to get vector slots. We've got our own supply in the UK now. We have done a deal with a company called Catapult, and we have our own dedicated vector supply from now on. So we are very well stocked in terms of vector. And the third thing we announced today was in the manufacturing of the cells is that we have successfully completed our first cell manufacture in-house in the Navy Yard in Philadelphia. So we're up and running with both in-house and out-of-house manufacture. So the next 3 years are going to be very exciting. On the partnership fronts, we have a lot of meetings this week, so I'll say no more.
Easy time for you, James. Good morning, everybody. My name is Pascal Touchon. I'm the head of Cell and Gene Therapies at Novartis Oncology. And as you know, I think the year has been very busy last year with Kymriah submission and approval as the first CAR-T approved in the US, and also the first launch of a commercial CAR-T in the US. But the year to come is even busier with a lot of activities, and since the topics around the next generation, I think we are already moving into a lot of new indication with Kymriah, and we're going to start more than 6 pivotal studies in 2018 with Kymriah in different type of CD19-based B-cell malignancies. We're also going to move into the multiple myeloma stage with BCMA CAR-T, so it's going to be very busy from a clinical development point of view. At the same time, we are globalizing Kymriah and moving into other territories than the US. As you know, we've announced that we've submitted the file for both the LBCL and pediatric ALL for Kymriah in Europe, and we're going to move in other geographies as well. So very busy time for the point of view of clinical development and regulatory submission as well. And we are now fully set up to manufacture that globally for a full access of the patient, which is our main objective worldwide, progressively in different sites that could accommodate this type of sophisticated and transformative therapy. What we're doing as well is to increase our research effort in terms of new construct, new technologies, and the ability also to do combination. I think there are a lot of combinations that are possible that have not been yet started, combination with other type of IO therapies, checkpoint inhibitors and others, and that will be able to make sure that we can make the T cells more active at some stages of the disease, and that will be certainly a very interesting and exciting aspect to watch in the next few months about combination therapy with CAR-T and Kymriah in particular. But beyond that, we're also looking at a lot of partnering initiatives, since we believe that this is just the beginning of the CAR-T era, but beyond CAR-T, other type of cell therapy in oncology, like what James and his team are doing on TCR, as well as some other type of cells, from NK cells to gamma delta T cells. So we are really looking forward as a leader in this field to do more partnering with biotech companies, academic centers, and to do much more into this field, because the medical need is immense, and the need to address solid tumors as well as other liquid tumors is great. So maybe I'll stop there, and we'll have a debate later on different aspects. But very exciting time indeed, and more exciting, very exciting time for the patients, because the fact that we've been able to treat commercial patients in the US is great, and shows that it's possible to deliver these therapies to many patients in different centers, and we look forward now to treat and hopefully cure many patients all over the world. So thank you for that.
Thank you. Scott Wolchko, I'm the CEO of Fate Therapeutics. At Fate Therapeutics, we're trying to pioneer a new approach to cancer immunotherapy, and we're trying to advance the field to move away from patient-specific and more towards a donor-derived, and ultimately a cell line-based for creations of cell therapy. Our first products we have is Fate NK100. Fate NK100 is a donor-derived NK cell therapy. It's in 3 different clinical studies. 3 INDs are open, 1 for relapsed refractory AML, a second study for recurrent ovarian cancer, and then a third study, which we're really excited about, that is just starting. We're combining NK cells with FDA-approved monoclonal antibodies for solid tumors, for instance, Herceptin and Erbitux. And so we look forward to generating clinical data with NK100 on those 3 different settings throughout 2018. What we're spending a lot of time on at Fate Therapeutics is actually pioneering this brand new approach to cancer immunotherapy. It's this notion that you use induced pluripotent stem cells. And induced pluripotent stem cells, I'm sure most people in the audience understand what those cells are. It's the ability to take a skin cell, for instance, and reprogram it backwards in life to a stem cell. And what's really exciting about that is that stem cell, induced pluripotent stem cells, can be indefinitely renewed. They can be differentiated into almost 200 different cell types in the body. But what's really, I think, powerful about the technology is once you create that cell, you can bank that cell. And you can engineer that cell and bank that cell. And what that means is, in effect, is you can essentially engineer once, create a cell, select a single clone. So now you have what's, I guess, analogous to the monoclonal antibody world of production. You essentially have a CHO cell line. And from that cell line, if you know the keys to the differentiation kingdom, if you will, you can create off-the-shelf NK cells, T cells, gamma delta cells in a single manufacturing run. You can create hundreds, if not thousands, of doses. And so at Fate Therapeutics, we're spending a lot of time pioneering that. We believe we'll be the first company, at least to our knowledge, to file an IND on an iPS-derived NK cell therapy. Our goal is to file that IND in the first quarter of 2018. As I mentioned, I think to our knowledge, it'll be the first ever iPS-derived cell therapy filed with the FDA in the United States, so super excited about that. And behind that, there are 2 other INDs that we expect to file in 2018 for iPS-derived NK cell therapies.
Okay.
We're working with the University of Minnesota closely on iPS-derived NK cell programs. That's being led by Jeff Miller.
Excuse me, Scott, can I get you, we'll get back to your specific trials in the next. Let me just move on to the introductions.
Sure.
All right, good, thank you.
Bob Azelby, the Chief Commercial Officer of Juno Therapeutics. Obviously, Juno's lead product is JCAR17, also known as Lysocell. We expect to file that product in the second half of 2018 in relapsed refractory diffuse large B-cell. And then we expect to move into earlier lines, in second-line non-transplant eligible and transplant eligible. CLL trial is open today, and then we're actively moving into BCMA and multiple myeloma in the early part of 2018. As Pascal pointed out, the combination opportunities going forward seems to also be a great opportunity because some folks, some patients, they get a response, but then the responses don't hold, but the CAR Ts are still there. And so we're looking at opportunities in combination to see if we can regalvanize those CAR Ts to have a big impact. As the Chief Commercial Officer, big focus for me is, understanding the market landscape and how Pascal and his team is going forward in the marketplace. We'll be third in the marketplace. So understanding the reimbursement landscape, how the providers are utilizing the CAR Ts, how are they getting reimbursed, what type of risk arrangements are going on, and how the whole reimbursement landscape is going to shift here in the coming years is a big focus of ours. And so, an incredibly exciting time, and I look forward to the discussion today.
All right, thank you. Thank you to the panelists. So the clinical outlook continues to advance for liquid and for solid tumors. Where do you see over the next 1 to 3 years the greatest progress being made? And what technologies, what therapeutic advancements are required to get us there?
Well, since we're in solid tumors only, I'm guessing that our products are going to be the greatest advances in solid tumors.
That's a softball.
We did have great data in synovial sarcoma. We are in 8 different solid tumors, and I think we're going to see what happens over the next year to 18 months in all of those solid tumors, because once you get it to an effective dose, there's a threshold with most of these T-cell therapies, and we think the threshold for us is about 1 billion cells. You should see efficacy, pretty quickly. But I would also echo comments made on my left that I think combination therapy is incredibly important. We certainly are absolutely determined to use combinations. But I think it is worth saying that there are 2 types of combinations. One is a combination with another drug, which I think is very, very important. And we ourselves, we have a combination study in multiple myeloma with Keytruda running right now. But I think the other type of combination you're going to see in cell therapy is where more than one thing, more than one construct is put back into the cell. We call those second or third generation, and we have a whole range of second generation molecules actually ready right behind the first generation ones. And I think that's going to be an area of great interest, exactly what the other speakers were saying. You still have the cells there, but for some reason the cancer comes back. And we think there are a number of strategies of putting something else into the T cell that can address that. And we think that's going to be a very exciting field over the next 2 to 3 years.
And on our side, we have 2 programs with our collaborators, collaboration with UPenn in solid tumors right now, one in glioblastoma with EGFRV3, another one in various solid tumors with mesothelin CAR-T. And it's still the early stage, I would say. It's good to be in a clinic and to learn from that first experience, but still the early stage. We're very confident that we will find a way for CAR-T to be able to address some solid tumors. There are some challenges regarding the target, regarding trafficking of the cells, regarding the immunosuppressive environment in the tumor. But we think that for combination and the right type of targets and construct, we can address this. And also we're looking at different type of other cells that might be more amenable to trafficking and addressing solid tumors specificities there.
I'll just add one point to that. There's a lot of talk about solid tumors and different indications, and all that's extremely exciting. But I think as we move forward here, we're starting to understand more about the type of cells that you put in the body matter. And so even in, whether it be relapse refractory, diffuse large B cell, or CLL, I think as the science evolves, you can understand, what are the right cells, what's the right mixture of cells in order to maximize efficacy and minimize toxicity. And so I think just in the early indications, I think there's a lot to learn there as well.
Yeah, I mean, I think in general, the success, I really can't predict where the success is going to come from, but I think that all of us probably agree that approaching the improvement or the strengthening of the CAR-T programs or the TCR program or whatever program you have in adoptive cell therapy is going to be dependent upon 4 things, some of which were already touched upon. One is, I think, in terms of CAR, further engineering of the CAR molecule is going to be important, and you mentioned the potential for dual CAR constructs, and there are other ways to engineer the CAR molecule. I think engineering the CAR-T cell itself, there's a lot that's being worked on now in some early stage companies that we're looking at that you may be involved in as well in terms of further engineering the CAR-T cell to address issues related to tumor microenvironment and other challenges, especially in solid tumors. And CRISPR is one way to do that. The third area is combinations we touched upon. That's the third bucket. And the fourth bucket is really something that none of us are going to be really excited to talk about because it's a lot of proprietary know-how that goes on in the cell manufacturing, the cell processing. You mentioned T-cell selection, or you alluded to T-cell selection. There's a lot of other things that go on in cell processing that we really can't talk about. But I think it's hard to say which one of those 4 buckets will emerge as most important. And it may be elements of 2 or 3 of those that are really going to combine to create a huge impact over the next few years.
And maybe to build on that, I think certainly in B-cell malignancies, we have now clear evidence of efficacy and manageable safety, which is leading to 2 approvals last year. But then we have still a lot of B-cell malignancies that needs to be approached for improvement in both the construct of the CAR-T as well as the combination and the manufacturing technologies to make that faster, more scalable, and cheaper at the end of the day. But then this is just part of the field, and we are very convinced that learning through that first generation of both indications and construct and cells, we can then apply many of the learnings to other type of indication target and cells. And we're very excited that the next few years are going to show a lot of excellent clinical results and hopefully save many lives.
Yeah, and I would just add, I think our focus is definitely on combinations. We tend to focus on FDA-approved agents that are already approved in combining cell therapies with those agents, like checkpoint inhibitors and like monoclonal antibodies. You can't deny that the checkpoint inhibitor space is just, expanding rapidly, as well as monoclonal antibodies, especially for solid tumors. And I think there's a huge opportunity to combine those traditional agents with cell-based therapies.
So with this
just to pick up something that Pascal's just said, actually, I think it is really important, this question of learnings from the clinic. I think if things go according to plan over the next 2 years, there's going to be an explosion of the words translational medicine and correlative data. I think it's going to be what actually happened to the cells in the patient, which ones got there, which ones activated, which ones actually did the business, and which ones were unnecessary, I think is going to be fundamental to understanding it. We ourselves have put in a massive effort into that, and I think those are the things that I would be looking out for at medical conferences over the next 2 years, is to see what actually happened to which type of cells and what type of cells did they turn into and which ones were effective, and did the companies learn about that and then feed that back into the clinical program. I think it's going to be vital to who succeeds and who doesn't.
So as we see this progress, and we're launching off of a great year in 17, and we see the tremendous expected growth that we'll have over the next 1 to 3 years, this issue of scale that was mentioned a couple of times during the last round, can we scale? Can the manufacturing keep up at this point? The accelerated approvals have put the CMC slightly behind the 8 ball in many cases now. And the question is, how can we catch up? How can we move ahead? And what are those logistic and manufacturing challenges that you see the biggest concern with, and how are you looking to solve them?
I think on our side, we are not that concerned about it, because we've demonstrated in our global studies that we could treat, within the current manufacturing technology, we could treat patients from Australia, Japan, Europe, the US, and other countries from a single manufacturing site. Whether that's going to be the future is another question. But at least this demonstrated. And I think that's very important, because that allows us now to move into regulatory submission all over the world in key countries and key sites, to be able to offer patients that treatment in a way that makes them very well treated, and hopefully to contribute to the transformative type of cancer care that we are really aiming at. But then, of course, this technology will evolve. And if we think 5, 10 years or whatever time, it will be more and more scalable, lighter, cheaper, faster. And we don't know exactly what's going to be there. But what we know is that today we've demonstrated that we have robust manufacturing technology that allows to treat patients worldwide, and then progressively this will improve tremendously. And we're going to be able, not only at Novartis, but I'm sure many of our colleagues in the industry, be able to invent new ways to address these scalable issues, or scaling issues there, regarding the ability to treat patients all over the world in different sites there. But I'm extremely confident that we're going to be able to do it immediately with the product that we have, and then progressively over the next few years with new technologies.
Yeah, just to add on that point, and I agree with everything Pascal said, I think the industry will evolve as well. As the science evolves and the excitement and the number of companies that enter the market evolves, you're going to see the supply chain become more mature, because right now I think there's limited supply in certain areas, so that drives up cost of goods and those types of things. And so as in standard business, as more things evolve and more competitors get in the marketplace, you're going to start to see COGS go down, automation get involved, and I think, as Pascal talked about it, we'll be able to scale these products.
And I think eventually you're going to see the model of centralized manufacturing, centralized cell processing kind of disappear. I can't say when that's going to happen, but I think it's just a matter of time, whether it's 2 years, 5 years, or longer. It is going to happen. There are companies that are already working on that. We're all aware of them. None of us, I don't think, has adopted them yet, but I think that's really going to be the future, and that's going to significantly address the issue of scalability.
From our point of view, if you just look at the position 5 years ago to now, first of all, the in-house process development changes that we've made to make it a practical proposition to make both vector and the cell manufacture, but also, as mentioned, the supply chain. It is exactly, I'm old enough to remember when monoclonal antibody companies built their own factories because they couldn't outsource supply. And exactly the same thing is happening in cell therapy. There has been an explosion. These meetings used to be 20 people around a table. We knew each other. That it is completely changed now. We can outsource many factors, but also we ourselves have, even just in the last 6 months, for example, we did something which quadrupled the yield out of the vector under certain circumstances. That's the sort of thing that's going to happen over and over again, and then it'll become, I think, routine and non-centralized.
Yeah, I would just add that I think absolutely the technology and the approaches today are definitely going to improve. I would say one thing I would consider as we think about next generation products, there will likely be multiple engineering steps that take place. And when multiple engineering steps take place, I think you are potentially going to need a different approach, a centralized approach, and a way to really qualify potentially at a clonal level, what engineering events have occurred so you can ensure you're delivering a safe and homogeneous product to patients, from patient to patient.
I think at the end of the day, they are still cells. So much more complex than a protein, which are much more complex than a small molecule. So whatever we do is going to need to address quality, as you said, as well as a very good interaction with regulatory bodies, because they want to make sure that what we deliver to patients is of highest quality and is reliable. And this dialogue that we need to have with them about how to define reliability in terms of a population of cells. And I think it was said earlier on how we can have a better biological insight about what's happening to these cells. The more we progress there, the more we'll be able to simplify our various processes and be able to deliver a product. But at the end of the day, these are still cells, so they are still very, very complex biological organisms.
As we saw in the stated in the briefing package and the industry report, the companies are now breaching geographic boundaries like never before. There are more companies developing in different markets, and each of the companies that is developing is looking to other markets as well. So there's and if you look at China, for instance, with the significant number of CAR T products that are coming out of China, just that one sector alone, how does that impact this regulatory question, and what's the regulatory environment look like, particularly as we start to look at this as a global industry, not a set of individual markets?
Maybe I start because we have some regulatory experience already. I think that it's been a very interesting relationship with regulatory because they didn't have any particular clue at how to handle this type of product, or whether it was even a product or a process. And I think all this worked over by yourselves and our colleagues in the industry over the last few years in the US, in Europe, as being very helpful to start to establish some key fundamentals about the regulatory perspective regarding CAR T and beyond CAR T cell therapy in oncology in particular. I know that there have been also some other activities outside of oncology. What's interesting in China is that there was a particular situation there due to the regulatory situation where CAR T in particular and cell therapy were not considered as product by the Chinese FDA till very recently. So that created the possibility to do a lot of studies at the hospital level, or even small companies setting up just to do studies, because that was considered as a procedure, so it could go very easily without having to go through the Chinese FDA CTA process there. As you know, this has changed now since December 22. There have been new guidance from the Chinese FDA there that is defining the cell therapy as a product that requires to be approved to go through clinical studies, that is approved by the Chinese FDA through the CTA process. So that's going to change the perspective of this field in China. But certainly what was good to see is that all this ability to do many studies has been also creating new ideas, new innovations, and that's been very good probably for the field. So let's see now how is that evolving. I think I would see, at least I don't know whether my colleagues agree, more harmonizations progressively. I mean, China has joined ICH as well, between the US, Europe, China, and other regulatory bodies from Australia to Canada, having a rather common view over these cell therapies, which is going to help us altogether to be global and not to have to do very different type of file for different authorities.
So we don't have experience in China, but I have to say that I've never known an industry get such a positive backing from regulators as cell therapy. Bear in mind, it's an industry where even the participants, you can't do animal models that really reflect safety, that there have been multiple deaths in almost every company which are unexplained. This is very difficult biology. The starting material for the manufacture is different because it comes from different patients. There are enormous numbers of things, and yet the regulators, both in Europe and here, have taken an amazingly positive attitude. We have breakthrough status here and prime designation in Europe, and the dialogue to try to get these things moving forward with the regulators rather than just something against the regulators has been astonishing. I mean, when we started, the RAC committee was incredibly important, sort of fundamental, sort of, could you actually put your product into man? That's really gone down in terms of the sort of importance. I would say that the support of regulators has been extraordinary for an industry in this stage.
So So let's move from the regulatory environment and the manufacturing, et cetera, to, we talked a little bit about reimbursement. We won't talk, maybe we can touch on reimbursement, but I'm particularly interested in hearing your thoughts on partnership and where Big Pharma comes in. You may know something about that, Pascal. And where is it how is this changing the industry now? What can we expect to see in terms of the big companies getting involved in the space as we go down the next 1 to 3 years?
As the only so-called big pharma in the room, I mean, at least on the panel, maybe I can comment on that. I mean, it all started with partnering. And we jumped back in 2012 with UPenn based on just a few patients, 3 patients being treated. And we thought it was a great innovation that we should try to bring to the market. And it's just 5 years we've been able to work very closely with our collaborators there at UPenn to bring that to the first approval. So it all started with partnering, and I think it's going to continue with partnering. I think what we just discussed regarding the number of questions that we have to solve, the number of challenges that we have in front of us, this can only be done through partnering. And I think there are many, many academic teams and small companies that can bring unique features to this field in terms of, again, construct technologies, facilitating the supply and logistics, as well as making sure that we can deliver at scale to many patients around the world, and then also doing this combination type of studies that we discussed, or dual construct or combined construct there. So the field is immense, and I don't see even a company like Novartis being able to do everything by itself. So we're going to rely a lot on partnering.
Yeah, so obviously from an in-licensing perspective, our lifeblood is partnering. Our 6 programs are partnered. The CRISPR program is partnered with Harvard. So, I mean, that's clear. I think what you're referring to is sort of out-licensing, partnering with larger companies to co-develop, co-commercialize our programs. And I think that's going to be critical to us. I think as we look around and thinking back to the sort of checkpoint story, where we evolved from a situation where at checkpoint there was a CTLA-4 antibody and that was it, to a point where it seems like every company, small or large, must have a checkpoint inhibitor of any ilk. We're far from being there with respect to CAR T. From this perspective of Mustang Bio, we love that to be the case, but it's not. It's clearly not. And so I think that it's really right now for small companies, it's kind of for a wait and see approach that we're seeing with respect to large companies, where people are not sort of jumping in left and right to in-license CAR T programs. Nevertheless, I think with the progress that all 4 of us, and I exclude you, Pascal, all 4 of us are expected, show in the coming years. I really think there's going to be a momentum to the point where, in a very short time from now, there's going to be a much larger demand for partnering with small companies with innovative adaptive cell therapy technologies.
I think we're already at the stage where it's become mainstream. I mean, if you count the big bio companies, obviously Celgene, Gilead, Pfizer with Selectis, GSK with us, obviously Novartis. As I say, if you came to these meetings 7 or 8 years ago, I think Pfizer was there because they had Selectis, and then it was us and about 3 other people. And I think it has become mainstream. I can tell you from the meetings that we have, requested this, people have requested from us this week. There are a lot of companies which are clearly interested in thinking about cell therapy. I think it's gone mainstream with the big companies now.
I agree. I mean, you see J&J with the Nanjing Legend situation. So Amgen started their own CAR T program in small cell lung cancer. I think this marketplace has evolved so much that every player in the industry that's excited about innovative therapies is going to have a play here.
And we're seeing a lot of this come out of Japan as well. The big, some really big companies. What do you think the impact of that has been, and what will it continue to be?
Well, Japan's had a surprising number of cell therapies in the clinic for a long time, actually. A company called Takara, I'm not sure if people are familiar with, has had several programs going for a long time, and they've had a very positive regulatory and clinical attitude to it. And I think that's sort of reflecting back into the big companies. We've always had Japanese interest in the TCR technology, actually, from, I'd say, more than from the US many years ago, and there's still interest in Japan. I think there's still a lot of interest in cell therapy.
Yeah, and on IPS, I think the Nobel Prize was delivered to a Japanese scientist there. But I think there are also in Japan a lot of institutes. We have a partnership with FBRI, which is an institute in Kobe, for tech transfer and local manufacturing, which shows that there is really an appetite there, not only at the level of pharma companies and biotech, but also academic institutions to be able to develop further cell therapy in Japan. So it's moving very fast there.
Okay.
Yeah, you alluded to it. I mean, given that Shinya Yamanaka was one of the founders of iPS cell technology, I think there's a tremendous amount of interest in iPS-based approaches to cancer immunotherapy coming out of Japan. Absolutely.
So let's finish on reimbursement then. We've got a few minutes left and talk a little bit about how you see this environment of reimbursement. Last year, as I mentioned in the opening remarks, we weren't quite sure how all of this would play out. There were companies having CMS discussions and that we weren't aware of what was happening. The alliance itself was in conversations with payers. But it looks like we're finding some creative ways to pay for these drugs. So what's left to do here?
Many things. I think it's so new and so transformative as a therapy that we couldn't just go there with a traditional way of looking at access, which I think is a key issue. The key issue is not pricing, it's access, and making sure we can organize the access of patients in the different geographies, starting with the US, where we are getting regulatory approval there. And so it was very nice to see that many part of the US system, and first and foremost CMS, were able to engage into a dialogue with us and find ways to address the unique features of that type of transformative therapy. First and foremost, the fact that this is a one-off therapy, and that this then creates a specific challenges on the system that is probably more attuned towards chronic disease and regular therapies used for years by patients, and a budget, yearly budget type of situations there. So that has been one of the first interesting aspect of moving into that field, which is how progressively open the various players there, be they commercial players or governmental organizations there, could really look at that with different eyes, and how can we work and engage in constructive dialogue to find ways to get patient access there. So we ourselves at Novartis very pleased in the way this is starting, and we have a lot of work to do then. And we're also pleased by the way we've been able to introduce Kymriah for pediatric ALL, albeit a very rare disease. Unfortunately enough for these kids, it's only a few hundred kids a year in the US. But it was very important that in making that commercially available, we were able to manage access for these kids. And we didn't want to have situations where we could not get access to the kids because of some issues with reimbursement or with the payers. So we've put in place various programs, and so far we've been able to deliver the drugs. And I think that's the most important at the end of the day. On the pricing aspect, maybe to comment, I mean, we wanted to make sure that we had a price that take into account the value of the therapy, the value for the patient, the value for as a medical value, the value for society, and the value for the healthcare system. And as you know, we've done a lot of cost-effectiveness analysis, and there were some presentation at ASH on what we've done there. We also had other type of organization like NICE in the UK or ICER most recently that have done with their own methodology, a different one. They've done also cost-effectiveness analysis, and all of these, whether it's internal Novartis analysis with external expert, or NICE or ICER, have confirmed that at the price we set for Kymriah, we are indeed cost-effective versus standard of care. And I think that's something important, but it's not sufficient. And that's why we in fact price the Kymriah at a lower level than the cost-effectiveness or the standard of care cost treatment, because we wanted to make sure that we have something that is, of course, in line with the value we're bringing to society, to the patient, to the system, but also in line with the investment that has been made and the need for the field to be able to further develop and get a proper return on investment there. So it's just the beginning, and there will be a lot of further debate regarding what's the level of pricing, reimbursement. But again, the most important has always been access and making sure that patient can be treated.
I think there's an issue of complexity in the pricing and reimbursement that that's easy to overlook today because right now the two therapies that are approved are essentially single infusion and then it's done. There's no reinfusion. As you look to the future, and I just cite the single case report that we had with respect to glioblastoma, where the patient got 16 infusions. It was a single cell processing event, but 16 infusions. I think that that's a little bit of a proof of concept and maybe a window into the future where multiple dosing is going to be important for solid tumors and maybe even for liquid tumors, and I specifically call out lymphoma, where there may be an advantage to reinfusion. Maybe there's T cell exhaustion, and there's some way around that where either the patient can be apheresed again with the same and produce the same product, maybe a different product. So I think that as we move forward in the field and we advance technologically, there's going to be the sort of dosing issue is going to be becoming increasingly complex. And so I think the way we're thinking about pricing and reimbursement today is probably not going to be the case in many instances going forward.
And just to add to that, I think on the reimbursement side of things, and a lot of talk these days on value pricing and risk arrangements, that there's a lot that goes into those, right? And so you have to have the right details. You have to, when you're setting up a risk arrangement, you have to make sure you're being measured on the patients that were actually done in the clinical trial, so you're doing apples and oranges. And then the ends that are being run in our studies are still relatively small, whether it be 75. So when you think about the long term and people are like, hey, just go at risk, there's a lot of details that need to go into it. There's a lot of tracking, baseline, and then making sure you settle up on the back end. And then the government with best price, there's a lot of mechanics that need to go into that so you can actually execute it over the long term. And so I think we'll continue to evolve in this space, but I don't think we're any close to being there right now.
Nothing, James?
I'm just delighted not to be the first to have to address it.
So we have a couple of minutes left. I'd like to just open up, if I could, to the audience. Are there any questions anyone would like to ask of the panelists? There's a first hand up.
I want to know more about any business development plan or development progress with autoimmune disease.
So progress on autoimmune disease? Anything?
At this stage, we don't have any program on that, but I know there have been a lot of companies working on Treg there, and certainly it's an approach that should bring some interesting development. I don't know whether anybody on the panel has...
We do have an autoimmune program. It's a preclinical program. It's based on the same concept that you could have a higher affinity T cell receptor, obviously different type of T cells, Treg rather than a killer T cell. That the concept is that you would actually be able to dampen down the effect. I have to say the targets are not nearly as clear cut as they are for cancer. They're much harder to find, and I think also what you're trying to do is something very different. It's much subtler. You're trying to alter a balance rather than kill something. So there are different challenges, also different manufacturing challenges. They're not as similar as I think they appear on the surface. You could just have a T cell for one and a T cell for the other. Actually, the programs require quite a lot of different technologies, both in the manufacturing phase, in the target phase, and in actually what you're trying to do. So we're very excited about the program. We have but it's a preclinical program. It won't hit the clinic for at least 2 more years. We're interested in the concept, but there are quite a set of different challenges, I would say. And I don't think at the moment there are targets for every autoimmune disease in the way that there may be targets for a lot of different cancers. We're finding the targets much harder to identify specific for an autoimmune disease because obviously the tolerance for off-target effects it may be lower even at the Treg. So we're cautious. We think it's very exciting, but we're cautious.
In December, Caladrius Biosciences completed enrollment of a 110-patient trial for diabetes, a randomized phase 2 trial using Treg cells on a platform T-regulatory trial. So we'll expect readout from that soon.
The advantages for off-the-shelf CAR T are obvious for the companies, but what are the advantages for the patients?
I think the biggest advantage for a patient is that when they come in, the doctor can immediately put them on the program. And with late-stage cancer patients, that's very important. I think it's less invasive, obviously, for a patient. I think there are all sorts of advantages. The whole patient experience would be different. You'd see an oncologist. You could be lined up for you'd still have to have the lymphodepleting chemotherapy, and then you could have the T cells the next week if that's what was suitable for the treatment. But you also don't need to go through the apheresis. So I think it's a much better patient experience if you can get that.
Yeah, because we are dealing with progressive disease, so the faster you can get the treatment of the patient, the better. On the other hand, what is the promise of CAR-T in particular and many of these therapies is long-term durable remission. Because that's why we believe there is a lot of value there in going into this type of product and therapies. So the key question will be whether this off-the-shelf allogenic type of cell therapy, can they really link to long-term durable remission due to the fact that most of the cells will just disappear after a while and will have not the presence of the memory cells that exist with autologous. And at the same time, the autologous field will, as we said earlier on, will make huge improvement in terms of the ability to deliver the transformed cells very fast back to the patients. So it'll be an interesting situations there. There might be also some tumors in which you don't want to have the memory, and you want just to have a relatively short-term, a few months type of presence of the cells to be able to address the disease there. So I think it's a very open field there, and there will be some interesting development. We at Novartis are looking at all the different type of technologies, and we have programs in allogenic as well because of that, because we don't know yet exactly where that type of off-the-shelves could bring not only benefit to the patient, but also address some specificities of some tumor types.
Okay, last word, Scott, please.
Cost of goods, multi-dose, dose titration, consistency of product patient to patient, single manufacturing run, thousands of doses.
Simple. So let me take the opportunity to thank our panelists for this morning for a great discussion. Appreciate it.